So good morning, and I'm very pleased to be with you. I'm in awe of the presentations that I saw before. I could not be with you in person, but that doesn't make it any less special. So I will take you out of the country to a cohort that many of you may not have seen before. I'm very thankful. I was one of the first CHEAR applicants and then transitioned into here. And the data that we're showing are very preliminary. We just got the results back late in the fall. And so this is a sneak peek, so to speak, of what we are seeing. I want to acknowledge my co-authors and co-investigators, Dr. Jeffrey Wycliffe, Arti Shankar, and Hannah Covert. And so today, what I'd like to do is introduce the cohort to you in general, provide you an overview of what is not metabolomics, particularly looking at the non-chemical stressors, and then share with you some preliminary results. And so where is Suriname? It's actually in the north of South America, if you follow that red line coming from Miami. It is important from an environmental exposure perspective because we're seeing by different geographic locations, different environmental exposures. And the best way to see that is through this slide. So if you follow along with me, the #1 location is the city, is the capital of about the whole country actually has 500,000 people. This is a cohort of about 1,200 mother and child diets, about 15,000 biospecimens, many still to be analyzed, and 16 questionnaires. And so the capital is one. The area #2 is the area where there is predominantly agricultural activity. And then the remaining are what we consider to be an interior. Of importance is #3 at that lake. That is where the mercury exposure is primarily happening through gold mining. And so the aims were fourfold, looking at a mixture of exposure to a mixture of developmental neurotoxicants, as well as non-essential metals, and considering both dietary, environmental, and occupational risks, and a series of biomonitoring efforts in pregnant women and their offspring. Then some environmental contamination assessments in fish, produce, and rice. Then focusing on bird outcomes and based on exposure in the mom of developmental neurotoxicants. And then lastly, taking it all together, including some neuroprotective nutraceuticals and going across the cohort as the children mature. The children are currently almost 6 years old. And so we started the cohort in 2012 with some preliminary analysis in 2009. Funded by the GeoHealth program. So this gives you a sense of some maternal demographics. It is quite challenging to do this kind of research in a country that is culturally very different. For example, we had to lower the intake rate or the inclusion criteria for pregnant women to 16 because of the cultural traditions of becoming pregnant earlier than what we used to here in the US. Ethnicity also quite diverse, as you can see. Maroons are descendants of African slaves, and they're now called tribal peoples, and a significant proportion of indigenous Native Americans as well. The education level primarily now or primary lower secondary vocational education, low income, and significant, I think, somewhat BMI, and then the 3 areas of the districts again. So if we take a quick look at the non-chemical stressors, the cohort and I should actually say that as part of this environmental epi cohort, 7 PhD in-country PhD students are graduating. Some already have been have advanced to postdoc positions and has received Fogarty Center fellowships. And so Anisma Gokul is one of those. And so what she has done is looked at both prenatal depression as well as perceived stress using respectively the Enberg depression scale and the Cohen perceived stress scale. So here are some of the data, and some that truly were surprising and interesting is that we saw at least a quarter of the women in first and second trimester self-reporting with probable depression, and same about half of the pregnant women reporting perceived stress using the Cohen stress scale. And so what we did is looking at depression across the various variables from social support to the perceived stress and educational level, and you could see the statistical significance both in the first and second trimester as well as in the third trimester. And so the non-chemical stressor exposure is of importance here. We transition into the chemical exposure. We're not discussing selenium here as the non-essential element, but that's one of the elements we have considered. So on the top of your screen is a very primitive device called a sluice box. It is in there where mud is assembled, and through a chemical process, mercury is released. The problem here is that the mercury, and to get out the gold, but the mercury is released in the surface water, so in creeks, in rivers, and in a lake where many of those descendants of Maroons live, work, and eat from the river as their primary source of protein. On the lower picture, you see Brazil nuts, and Brazil nuts are very loved by this population, by this subpopulation, and Brazil nuts are high in selenium. On the right-hand side is what we think is the source of lead. What the woman is preparing is kind of a pizza from yucca, from cassava, but she's preparing it on a kind of a pizza plate. It is a steel and an iron component, and we think that the lead is being released from that. But that's only one source of the lead. In terms of mercury, you might recognize this as the piranha. And so because of the practice of mining, mercury is released into the rivers and of course goes up to the food chain. And it so happened that the piranha is one of the highest contaminated with mercury, but one of the most loved in terms of fish for consumption. And fish represents almost the sole source of protein for the pregnant women living in that area. And so back to the map, you see the city again, Paramaribo, but it is that blue lake that we're worried about. It is in the area of that blue lake that actually streams back into the Suriname River, where those practices that you see on the right-hand side, in fact, on the upper right-hand photo was primary rainforest and disappeared within 90 minutes because I take students in the interior to show them what mining does to the ecosystem. And then so below that, you see how that primitive dredging and retrieving of gold occurs. I am not acknowledging here both the Patrick Parson, who's speaking tomorrow, the data that we just received from them on the metal data and our own metal data we did before. And as you could see by interior, by the regions, interior, the agricultural area and the city, so interior, Nikeri and Paramaribo, you see the levels anticipated. We have seen what we expected, far above the action level of mercury in blood in the interior, where the gold mining is happening. Same thing is for lead. But then for manganese, we see a higher level in Nikeri because manganese is a component of one of the pesticides that are used there a lot. Low levels of cadmium across, but some differences. Now, if we only looked at the Wadsworth data and looked at by trimester, by region, so location by trimester, same pattern, both for lead, mercury, and manganese, as well as cadmium. And so there's quite some consistency there. We have begun to, and we're about actually. One other PhD student has submitted data that will be published soon. It is in review of neurodevelopment, particularly using the Bailey scale. This is one of my co-investigators actually in Suriname for the Bailey testing. And here's some of the early data that we're seeing. And so if we took lead alone, stress alone, and probable depression alone, and looked across the different subscales of the Bailey, you see that some are statistically significant. And so if you take them together, that's the last row, the cumulative risk index, you see the levels that we are continuing to explore more. But what was important, if we took only lead and looked at exposure and toddler neurodevelopment, and we see that for levels above the 3.5 micrograms per deciliter, we see both the impact, a significant impact on receptive and expressive language domains. So now transition to the HEAR metabolomics. Very much thank you to Susan Sumner and her team. We had about 350 samples from 350 participants, serum samples, and by trimester, both late first and early second and third trimester. And so about 15,000 features came out of the raw output. 9,100 made it past QA/QC, and about 2,500 came out using the interquartile range analyses and metabo analysts as the database. I need to immediately tell you that I am not a metabolomics expert, and so what you're seeing is some very, very early peaks as we look at the data. And so the three groups that we're looking at, again, by the three regions, the interior, the urban capital, and the agricultural district, you see that some were non-significant, but the ones in red were all statistically significant using one-way ANOVA. Then, again, looking across the three regions and considering several of the metabolites, we have just begun to decipher what all these metabolites mean. But you could see that there are differences across the three regions, and these differences, our initial thoughts are that they definitely have to do with diet and the differences in diet, which you could see on the upper right-hand side, the three areas as well, against the different metabolites that we're considering. There are differences, as you can see across, and there's not a total overlap across the three regions. So again, indicating that diet plays a role. We also looked in the interior, just in the interior cohort, the sub-cohort, between first and second and third trimester, and doing some t-tests. We took one of these pink dots, so to speak, these pink metabolites, and looked further. One is the, again, we're just looking, but this is interesting to see that there are differences between the first and the second trimester and the third trimester. It's the one, K16, that's the one that piqued our interest, and that's diosgenin. It's as you know, it's a steroid. What's important for us and you see that it is higher in the first and then the third trimester. What's important to us is that it has medicinal qualities, and as I shared with you earlier, there is a significant cultural emphasis on medicinal plants and the use of medicinal plants, not only in daily diets, but particularly during pregnancy. And it is that, for example, many of the pregnant women, particularly of the Maroons, the tribal peoples, eat dirt, so clay, it's called pimba, because of the minerals in it, their consideration, their beliefs, and they're not the only population that does that. And so we're wondering how much is in that. There also is a very favorite jam that they like to use all across in Suriname, but including in this subpopulation, and they make kind of a chicken pie with it called POM, P-O-M. And so this is new territory for us to begin to look at the role of diet a lot closer as we're beginning to decode what all these metabolites are. This gives you another sense of looking at, again, across gestation for the interior. Now, you might wonder why we're focusing so much on the interior sub-cohort. It is because both lead and mercury are extremely high in that sub-cohort. It's also because their diet significantly differs from the other two subregions. So what's next for us? Looking closer at what we're seeing in terms of metabolomic profile and demographics, looking closer across gestation, not only for the interior sub-cohort, but for all three cohorts, so for the entire study population. Really cross analyses of environmental exposures to the different components of the mixture and individual components and the metabolites. And then particularly then focusing on the maternal and child health relationship, both in terms of diet, and one of the concerns that I did not mention earlier is that about more than half of the women in this cohort are anemic. And so how that plays in terms of the metabolomic profiles. Focusing on birth outcomes and all the variables that we have that we looked at for all the sub elements of birth outcomes. And then continuing to follow the kids now that they are entering kindergarten and first grade. And so a lot still to do. This was a sneak preview, and it leaves me with acknowledging the participants, of course, the CHEAR program, the funding coming from the Fogarty and from NIEHS on GeoHealth, all of our principal investigators and co-investigators, and particularly our former graduate students who are now postdocs, and many of them UGMT fellows. The Ministry of Health in Suriname plays a key role, and the research center in the academic hospital in Suriname, where all of our biospecimens are stored, and several analyses are being done. And leaving you in a multicultural mode. This was a celebration of an Indonesian holiday, and the last picture down there was the latest group of students I took with me, as I do every year, to not only experience culture, but seeing the impact of environmental exposure on pregnant women and children. So be pleased to answer any questions.